Hello and welcome to this webinar on extracting research evidence from publications. My name is Maria Levchenko. I work in the Literature Services team at the European Bioinformatics Institute. I'm responsible for outreach and training for Europe PMC, a literature database for life sciences. And I'm here today with my colleagues, Francesco Talo and Aravind Venkatesan. Both are our text mining and data science experts. Over the course of this webinar, we will demonstrate how to access text-mined literature evidence using Europe PMC Annotations API. We know that our audience may have different levels of experience in bioinformatics and programming. We expect that you have some prior knowledge of programmatic access. If you are a complete novice, don't worry. We will have some introductory stuff to get you started. We can also recommend viewing another webinar from EMBL-EBI for Introduction to Programmatic Access. For those of you who are quite advanced, please stay with us. We will cover more challenging things towards the end. If you have any questions, you can always contact us via the help desk at europemc.org. So, let's get started. Here's what we're going to cover today. First, we will explain what kind of data you can retrieve, including annotation types and sources. We will then move on to the API itself and introduce available tools and operations. We will briefly look at the different output formats to retrieve the data. And we will finish with a couple of examples, focusing on biological use cases. But first, let me tell you a bit of where we're coming from. As I have already said, Europe PMC is a scientific literature database. It provides access to biomedical research articles, patents, clinical guidelines, and other relevant documents. Europe PMC is a public resource. It is freely available to everyone without fees, subscription, or registration. It is also a designated repository for 28 life science funders. This means that researchers supported by these organizations can deposit their manuscripts in the database and provide access to the full text of research articles for everybody else. Europe PMC is a partner in PubMed Central International, sharing full text content with PMC USA. Like PubMed and PubMed Central, Europe PMC also has a web interface for literature searches. Where Europe PMC differs is that we combine both PubMed abstracts and PMC full-text articles in one search interface. We supplement it with additional resources and layer some innovative services on top. One example would be the Annotations API. At Europe PMC, we consider research literature an important part of big data in biology. There are currently over 33 million publication abstracts that are available in Europe PMC. That is 10 times the number of genomic variants in DBVAR database, or 240 times the number of protein structures available in PDB Europe. Journal articles are the major dissemination route for new findings. They contain facts and data that can be extremely valuable for future studies. Scientists access this evidence simply by reading. However, with the current publication speed, research on diabetes, for example, will produce about 70,000 papers a year. To scan through all of them, you would need to browse more than 7 papers every hour of every day without any breaks. To extract knowledge from such a large number of publications, automation is the key. Text mining is often used to automate knowledge extraction. However, its application can be limited because of the technical know-how to set up a text mining pipeline. At Europe PMC, we wanted to help bioinformaticians and wet lab researchers to overcome this problem. Therefore, we have set up a community platform for text mining outputs. This platform allows text mining experts to share their results with biomedical researchers by submitting text mining data to Europe PMC. We then make this data available to public. This means that you don't have to be a text mining expert to do literature analysis. all you need is a bit of programming knowledge and a good idea to apply this to. First, let's look closer at the data available on the platform. Question 1. How comprehensive is it? As I have already mentioned, Europe PMC contains 33 million abstracts, of which 28 million come from PubMed and 5 million from other sources. Of these 33 million abstracts, 4.6 million also have full text available to read. Although these full text articles can be freely accessed, their copyright doesn't always permit reuse, including text mining. Therefore, text mining data in Europe PMC is only available for so-called open access articles that are licensed for reuse. As of February 2018, there are 1.8 million such articles in Europe PMC. In contrast to that, research abstracts are not subject to copyright and therefore can be text mined freely. So the literature corpus that is covered in the Europe PMC text mining platform consists of all available abstracts and open access full text articles. Next thing you probably would want to know is what kind of data can be retrieved via the platform. The outputs of text mining are submitted to Europe PMC as annotations. Imagine a text mining group is interested to identify some concepts. Those concepts could be gene names or protein interactions or any other relevant pieces of information. Concepts found in the text are tagged or annotated. In a way, a text mining annotation is simply a label, telling us that in this sentence, P53 is a protein and methotrexate is a chemical. So, which annotations are available? Currently, there are 8 annotation providers sharing their outputs via the Europe PMC platform. The list includes the NCBI, the Swiss Institute for Bioinformatics, National Center for Text Mining, and so on. You can see all of the annotation providers on this slide. Each of these providers submits a different type of annotations. And here is the list of available annotation types. I just want to highlight one of them, the accession numbers, which is an annotation type for research data. Accession numbers are unique identifiers. They're given to a data record to track it in a repository. For example, 2BK9 is an accession number for a record in PDB Europe database that contains structural coordinates of the globin protein from Drosophila melanogaster. Now, coming on to the API. There are actually 2 ways in which you can access annotations in Europe PMC, via the RESTful API or on the website as highlights in the text. This webinar will focus on the programmatic access to annotation data, but if you're interested to know more about Annotations app to help you skim read an article and locate primary data quickly, you can watch a video tutorial on searching for research data in scientific literature. You can see the link on this slide. As for the Annotations API, you can find it on the Developers section of the Europe PMC website. I have put the base URL here for you. To see which tools are available to you, let's focus on the API controller. The API has 5 different operations which you can use to retrieve text mine data tailored to your needs. Each operation performs a distinct function. For example, retrieving a list of annotations for a specific article. We will look at the operations in more detail in a couple of minutes. For each operation, you can specify several parameters to fine-tune your query. We will look at the parameters now, but you can always refer to the API documentation to check. There's no need to memorize them. There are 2 parameters that are important for navigating the results: page size and cursor mark. We define one result as one article that contains all instances of your annotation of interest. So, one result equals one article, potentially with many annotations. Results are arranged in pages, and the page size parameter defines precisely that, how many results you can see on a single page. The value that you can input must be between 1 and 8, with 4 being the default. You want to be able to navigate between different pages to retrieve results that weren't displayed due to page size. That's where you will need the cursor mark parameter. The cursor mark specifies the next results page. It's a kind of a programmatic bookmark. To recall the next page, just input the next cursor mark value that you can find at the top of your results list into the cursor mark parameter. Here's an example. That's the first page of my results, and right at the top I have the next cursor mark that I can copy and then paste here to go to the next page. Another important parameter is filter. Filter has only two values, 1 or 0. If the filter parameter is equal to 1, which is also its default state, for each article in your results, only the annotations that you have requested will be retrieved. So if you search for a specific entity, let's say P53, in your results you will retrieve a list of articles for each of which there will be a list of all P53 mentions and nothing else. If, however, you set the filter parameter to 0, you will get all the annotations for the same list of results. A bit confusing, so here's an example. If you want to find what other concepts are mentioned in papers on P53, you will search for P53 annotations with the filter set to 0 and get a list of articles which have at least one P53 mention annotated. And for this article list, you will get all available annotations, chemicals, proteins, organisms, you name it. The last parameter that I wanted to mention is the format parameter. You can use JSON, which is also the default parameter, to import the data into a web application. You can recall XML, which is commonly used for archiving, for example, to import your results into another database. For the data scientists among you, we do offer JSON LD for linking data from different resources. And finally, you can choose ID list. The latter will produce a list of article identifiers and will not retrieve any information about the annotations themselves. This will be a very useful format for some of the things that you can do, and we will show you a use case for it later. So now we have covered most common parameters, and we can get back to the list of operations that are available for the Annotations API. These are basically your tools, your methods, that allow you to answer different questions. Let's start with Get Annotations by Entity. It allows you to get the annotations of the article which have at least one annotation of interest. So how does it work? Let's say you're interested in research on Arabidopsis. It's a common plant model organism. In this case, you would input Arabidopsis as your entity of interest and retrieve all Arabidopsis annotations available via the platform. You should note here that an entity doesn't have to be a single word. You can of course also use a phrase, for example, cardiovascular disease or Staphylococcus aureus. The next operation is Get Annotations by Provider. As the name suggests, it allows you to get the annotations contributed by a specific provider. So if you're only interested in gene-disease relations that DisGenet has provided, but not gene-disease associations from the Open Targets platform, you can use this operation. Get Annotations by Relationship deals specifically with two types of annotations: relations between genes and diseases, and relations between transcription factors and their target genes. This module allows you to retrieve annotations for associations between two entities of interest. For example, P53 and cancer. So this means that you will need to input two entities. Again, those could be single words or they could be phrases. Now, Get Annotations by Section and or Type is a really cool function. It allows you to do two things. It allows you to retrieve annotations by article section and here we mean sections like introduction, results, or materials and methods. Or it allows you to retrieve annotations of specific type, like all annotations of chemicals. You can also combine them both to find, for example, chemicals mentioned in the materials and methods across all papers. We will come back to that use case later. So finally, we cover get annotations by article IDs. This method allows you to retrieve any annotations contained in the selected list of articles. You need to remember to input no more than 8 article IDs at a time. So your list should be restricted to 8 IDs. One question that you might ask is, how do I find the article ID? Now, an article ID has the following format: source, external ID. The source is a 3-letter code for the resource that has provided the data. There are several data sources included in Europe PMC. For example, MED stands for Medline abstracts that are available from PubMed. You can see a full list of data sources on this slide, or you can also view them in the parameter description. If you have a paper in mind and you would like to find its article ID, you can do so either on the live site using the web search or programmatically using the search module of the Articles API. For example, here is a paper in Europe PMC and I have highlighted its external ID. Since this is a PubMed identifier, the full article ID that you should use with Annotations API would be med, 29370,687. As you can see, there are several different tools available to use to solve different problems and answer various biological questions. Let's try them out and see what we can get. Let's start with a simple example to familiarize ourselves with the API response. Say you wanted to retrieve all mentions of chemicals from the methods section of full-text publications. We're already familiar with the get annotations by section and/or type operation. We will input chemicals as our annotation type and select methods as our section of interest. For those of you familiar with the RESTful web services, you will know that REST queries come in the form of structured URLs. You can see the response body directly in the controller, or you can use the request URL provided here and paste it in your browser. This will be a REST interaction even without any programmatic access as such, meaning that there's no need for you to write a code or use a specific client. So here what happens if you use that URL in your browser. You will see a result list that consists of 4 articles. As you remember, this is defined by the page size parameter that we left to be the default. If you expand results for one of these articles, you will see the article identifiers along with the list of annotations for the articles that we have retrieved. This particular article has 1,255 instances of chemical names mentioned in its methods section. Just to let you know, this is a methods article published in a chemical journal, so that checks out. Now let's look at what information is provided for each annotation. Prefix, exact, and postfix all define where the annotation is found in the text. Prefix is a text snippet found before the annotation, and postfix is a text snippet immediately after. That leaves exact to be the annotated entity. In this case, it's a chemical called calixaran. Now you will notice that here the prefix is empty, and that is because our annotation is actually at the beginning of the sentence. You can see an extract from the paper with the sentence at the very top. For some annotation types, an entire sentence is annotated. So there will only be an exact. There won't be any prefix or postfix. Now, your response also includes annotation tag. You might ask what it is. In order to efficiently share and reuse text mining data across different tools and platforms, we formatted according to the WC3 Web Annotation Data Model. This particular form treats an annotation as a link between the tagged entity in the text and the underlying data record. In this example, calixaran is a chemical. It has an entry in the chemical dictionary. And here, under tag, you can see a link to EBI dictionary. If you follow it, you will get to the calixaran entry in EBI database. Now we have explored how to use the API via the browser. But the real power of an API comes with combining different queries to ask more complicated questions. To do so, you probably would write a script that runs in a programming environment. The RESTful API can be combined with a plethora of programming languages. We will now show you an example of a Python client that retrieves gene mutations mentioned in papers on diabetes. Now, in order to construct this query, let's think what we actually want to retrieve. First, we want to know which papers discuss diabetes. For that, we can use annotations by entity and input diabetes. We will request ID list as our result type because we're only interested in the list of papers. Next, we would take this list and use annotations by article ID to retrieve annotations that we will limit to gene mutations type. In theory, you don't necessarily have to write a program to retrieve this data. You could use the logic we presented here and get results manually via the browser interaction. But we will try to automate it using a simple script. Now here is our Python script. Basically, it has two functions. Function IDs calls the base query URL, in our case, the URL of the Annotations API, and retrieves the list of article IDs that have an annotation specified as entity. In this case, entity equals diabetes. And function annotations will retrieve the annotations of the specified type from the list of articles that the function IDs has retrieved. Here's how the function IDs looks like. It basically constructs the URL that you will call the Annotations API, use the Annotations by Entity module, define the entity as diabetes, and output in ID list format. The output will be processed as article IDs and will be stored as a list. In the second function, we will call the Annotations by Article IDs module, limit the annotations to gene mutations with the type parameter, input the list that we have retrieved with the first function, and return annotations that we have requested. Here's how results look like. We have retrieved them in JSON format. You're already familiar with this layout from our previous example. Here the annotation for gene mutation is represented by an accession number, RS499974. Now, to be exact, our script is missing one important part. For simplicity, it only retrieves the default 4 articles. If you wanted to retrieve the full list, you would have to add a piece that uses cursor mark parameter in a loop and goes through all of the result pages. There are many more things that you can do with this tool. You can find out which potential treatment targets have been linked to a specific syndrome in the literature, or you can identify papers that cite proteomic data. You can combine Annotations API with Articles API to find out which components of a signaling pathway already have a protein structure available, or which studies report gene mutations and link to clinical trials. There is a lot of flexibility within this tool, and we hope that you will explore its potential applications. The last but very important part is on how to get help if you get stuck in any way and where to find more relevant information. If you're planning to use the Annotations API in your future work, I strongly suggest that you join our developers Google group for updates and discussions. You can also post your API-related questions there, and you can see the link to the Google group on this slide. If you want to contact us with any other queries, we do have an active help and support team at helpdesk@europemc.org. You can also follow us on Twitter to keep up with the new developments or learn about upcoming webinars or hackathons. The Train Online site has the full list of courses offered at the EBI. If you'd like to go back and revisit this webinar, it will be available on the EMBL-EBI Train Online website. Thank you so much for watching. I hope you enjoy.